Hello ladies and gentlemen, and welcome back to C.A.R. Entertainment. I'm your host Dylan Hamilton, and today guys we're going to be continuing with our AT2 biology revision video with guys, and in our inheritance area topic of AT2 biology. And last time we talked about the monohybrid inheritance. Now we're going to talk about sex inheritance and how you inherit what sex you're going to be or what gender you're going to be. So without further ado, let's just get straight into this. So our in-human sex determination is pretty simple because we inherit 23 pairs of chromosomes, so 46 in total, but one of these pairs is our sex chromosome, so XX or XY, as we see. So the other 22 that we know are called autosomes. If you're a male, you have XY. If you're female, you have XX. Females are called homogametic because they have the same, and then males are heterogametic because they're different. X and Y are different. So the inheritance of sex-linked characters. Well, these genes will be found on the genes that are found on the X chromosomes are sex-linked, and a person who is a hemophiliac or carries the gene for hemophilia will almost always occur in males only. Doesn't really go into females, and this is because it's caused by the presence of a single recessive allele on the X chromosome, and the Y chromosome is almost genetically empty. Now, this is what I'm going to explain. So hemophilia only always occurs in males because it's caused by this presence of a single recessive allele on the X chromosome. If you're a male, you only get your X chromosome from your mother. Okay, so that mother will have a single recessive allele, but if she has a recessive allele, it's not going to show because it's not going to be dominant. Therefore, they're going to have it from their mother. Okay, so the mother will not be hemophiliac because it's a recessive, but when she passes it on to the child, they'll have two recessives, and therefore they'll be a hemophiliac. And a hemophiliac, the hemophilia is a recessive sex-linked genetic disorder that affects the ability of the blood to clot. Basically, your blood doesn't clot. So, what are some other sex-linked genes? Well, color blindness, which is a recessive mutation found in the X chromosome, muscular dystrophy as well. In fruit flies, you have this wild type allele known as X+, and this will code for the red eye and is dominant. I think maybe X.R even, sorry. And then you have the opposite allele, which is allele XW, and this codes for the white eye, and this is a recessive one. So we go through things called pedigree charts, which are used to chart the inheritance of a condition or a trait through several generations of a breeding group. So things like the Huntington's disease is an autosomal dominant because the condition does not skip a generation. It allows you to pass through each generation. So what does autosomal recessive mean? Well, this means that the trait will only appear in some generations, so not all of them. As autosomal means even numbers in males and females. So autosomal dominant means that it is impossible to pass this condition on without at least one parent having the trait. And again, there's an even number of males and females. If it's an X-linked recessive, then it's more common in males but not present in all males. If it's X-linked dominant, then if dominant, it's not mainly restricted to male. Okay, so for example, your hemophilia is X-linked recessive, Huntington's is autosomal dominant because it's a dominant allele. So lethal allele combinations, and then we'll finish off. So this is probably the best way to go through and describe how lethal allele combination will arise from this. So as we see here, homozygous form Manx cat has a gene that's lethal, and the embryos will fail to develop, and therefore they'll die. So as you see here on the left, we'll just bracket the left side first, so you see your phenotype there at the left, and you have your Manx and the normal. So we're crossing the genotype of capital T, small t, and the normal should actually be two small t's. That big T, I couldn't really do anything about it. So the gametes, you're going to have a big T and a small t, and then for normal, you're going to have two small t's. So what we do then is we have this cross. So you have the two t's at the top, and then a big T and a small t at the side. So when you cross the big T with a small t, you're going to get a big T, small t. When you cross big T with a small t, you're going to get big T, small t. When you cross two small t's, you get two small t's. You cross two small t's, you're going to get two small t's. So you're going to get ending up with two Manx cats and two normal cats. And these genotypes mean you're going to get two big T's, small t's, and two small t's. So then, if we cross a Manx cat if we cross two Manx cats, so two Manx cats breed with each other, where the gametes are big T, small t, and big T, small t, we cross this, and you get the two T's cross with each other to get two big T's. The big T, small t gives you big T, small t. Big T, small t is big T, small t, and the same for the last one as well, which means we get two Manx cats and one normal, because the one that has two capital T's will not, because as we see, a homozygous form of the Manx, which means it's the exact same. As we know, the capital T gives the Manx cat that is what makes you a Manx cat, the capital T. And there, if you have two of those, your embryo will fail to develop and you'll die. So instead of having the 3-to-1 like we usually do, we have 2-to-1 because we have the capital T and a small t, and then the 2 small t's. Thanks for watching this video. I've been Dylan Hamilton from C.A.R Entertainment, and I will see you next time.